Hi, I'm Chen. In my project, I developed a computational pipeline to identify potential alternative splicing-derived neoantigens, and I applied my work in the context of acute myeloid leukemia. To start off, I want to explain what alternative splicing is. Inside cells, DNA is transcribed into pre-mRNA, which is spliced so that introns are removed and then translated into protein. Alternative splicing is the biological process that enables multiple variants of a protein to be generated from one pre-mRNA transcript. In cancer cells, abnormal alternative splicing can introduce novel, cancer-specific transcripts that can be presented through HLA class I molecules on the surfaces of cells, where the immune system can recognize them as foreign. These presented peptides have important immunotherapeutic applications because they're potential neoantigens, or cancer-specific peptides that may allow T cells to recognize and destroy tumor cells. I developed a pipeline to predict alternative splicing-derived neoantigens from tumor RNA sequencing data, or RNA-seq data, and to validate the pipeline's effectiveness, I tested it on the B721-221 cell line, which, in addition to RNA-seq data, had ribosome profiling and immunoproteomics data available for analysis. But let's start from the beginning. RNA-seq reads provide a rough readout of the mRNA transcripts present in a sample. My pipeline uses a transcript assembly tool called stringtie to reconstruct the full-length reads that RNA-seq reads are derived from. In broad strokes, the pipeline then identifies transcript isoforms with instances of abnormal alternative splicing and determines the peptides that they yield. Up to this point, the pipeline only considers RNA sequencing data and information about mRNA transcripts, but to predict potential neoantigens, we also need to consider whether transcripts are likely to be translated and presented by HLA class I molecules. To that end, we can leverage ribosome profiling data, which provides a readout of mRNA translation, and we can use a prediction tool called HLA Athena to determine if a peptide is likely to be presented by HLA class I molecules. If an alternative splicing-derived peptide has evidence of translation and is predicted to bind to HLA class I, we consider it to be validated. In the B721 cell line, I predicted 670 alternatively spliced transcripts that yielded about 160,000 peptides. I then validated 192 of those as both translated and predicted to bind to HLA class I, constituting my set of high-confidence alternative splicing-derived peptides. More recently, we've also performed an ad hoc immunoproteomics analysis to give us a picture of what peptides are actually presented via HLA. Preliminary results have validated 38 predicted peptides, 91% of which come from riboseq-supported events. Now, moving to results, I applied the pipeline to RNA sequencing data from 8 acute myeloid leukemia cell lines, or AML cell lines, and 7 AML patient samples. I focused on AML because, compared to other cancer types, it's been found to exhibit higher levels of alternative splicing, and one type of event, in particular, intron retention. So I detected an average of around 700 alternatively spliced transcripts across cell lines and about 1,600 across primary AML samples or patient samples. Now, ongoing efforts are directed toward generating ribosome profiling and immunoproteomics data in AML cell lines for further validation, and also toward determining what RNA-seq features are associated with a higher probability of translation and HLA class I presentation. Notably, compared to the B721 cell line, AML cell lines had a higher proportion of intron retention relative to other events, around 30% compared to around 10% in B721, which is in line with previous findings on the prevalence of intron retention in AML. Another relevant observation is that 31 alternative splicing events predicted in the patient samples are shared by all 7 patients, shown here. And while immunotherapies targeting mutation-derived neoantigens are, by their nature, patient-specific, alternative splicing-derived neoantigens have the potential advantage of being shared across patients, which could allow for universal, off-the-shelf immunotherapies applicable to different patients with the same cancer type. This would be particularly relevant in tumors like AML with a low mutation rate, for which identifying potential neoantigens remains challenging. So, stepping back, this work has explored alternative splicing as a source of neoantigens, covering ground from patients to neoantigen prediction. Next, I hope to validate the cancer specificity and immunogenicity of my predictions to verify that they are not shared by normal cells and are actually capable of eliciting an immune response, both of which would be necessary for their exploitation in T-cell therapies or cancer vaccines. Overall, this work brings us a step closer to designing novel cancer immunotherapies centered around alternative splicing-derived neoantigens. Thank you so much.